Alrighty, so we're into the third and final process of gene expression, translation. So this involves taking the mature mRNA now from our post-transcriptional modification and using it as an instruction manual to piece together a string of amino acids called our polypeptide, which will then fold into a functional protein product of the original gene. So now again, similar to transcription, this is a dynamic process, so we'll work through each of these parts and then kind of put it all together at the end. We start with our mRNA strand and a ribosome which reads the mRNA in its 3' to 5' direction, just like the RNA polymerase did in transcription. It's going to read the mRNA in groups of 3 nucleotides at a time. These are our codons. So each codon triplet will have a corresponding tRNA molecule carrying a complementary triplet code, which we'll call the anticodon. On those tRNA will be a specific amino acid. So we always start with the codon AUG, which brings in a tRNA carrying FMET, or short for formal methionine, and then we move on to the next codon in the mRNA molecule. We'll get another codon, which will correspond to a different tRNA carrying an amino acid, and this will bind again, and the new amino acid will form a peptide bond, seen here, to the FMET to form a dipeptide. So this will continue, and the old tRNA will detach and be reused as we go while the polypeptide sequence will grow in length. So we eventually get to one of 3 possible stop codons, which brings in an empty tRNA carrying no amino acids. And this is the signal for translation to end and the polypeptide to fully detach. And that's it. So now to structure these steps, we can again break this process down into the initiation step, right? Just like we did with the transcription. So the ribosome reads the AUG start codon and recruits an FMET carrying tRNA. Then the second part is the elongation step, which we talked about, which involves the bulk of the reading and the amino acid recruitment. And then finally the termination phase, where the ribosome meets one of these 3 stop codons and the polypeptide detaches. The polypeptide is now free to begin its folding process into a secondary, tertiary, or even quaternary structure, meaning we've successfully used the original gene from our DNA to encode and manufacture a fully functional protein through gene expression. Cheers, guys.